Hello, everyone. Thank you for attending my web seminar about selective brain region vulnerability in Parkinson's disease and how this is affected by local variances in alpha-synuclein conformations. To give you a little bit of background, the talk will be going mostly about a group of diseases called synucleinopathies. There are various neurodegenerative diseases that have one common denominator, and that is Lewy Body Pathology or Lewy pathology. Lewy bodies are found in Parkinson's disease, dementia with Lewy bodies, and multiple system atrophy, as well as in the amygdala of 50% of Alzheimer's disease cases. The pathological hallmark of these are protein inclusions. As usual for neurodegenerative diseases, you'll find protein inclusions in the brain, in the affected brain areas of the patients. And these will consist of a specific protein. In this case, as the name synucleinopathies already infers, the protein is alpha-synuclein, a small presynaptic protein that is actually normally expressed at high levels throughout normal life. There are a couple of implications why this protein might be causative in these synucleinopathies. One is, as I already mentioned, the pathology that mainly stains for alpha-synuclein, these protein clumps that you find in patient's brain. But also there are 5 published point mutations in the alpha-synuclein gene that are associated with early onset of developing a synucleinopathy, as well as very high penetrance of developing a synucleinopathy. Another genetic hint we also got that people who express higher levels of alpha-synuclein, so duplication, triplication, or quadruplication carriers of the SNCA gene, are actually also very likely and with high penetrance probable to develop Parkinson's disease or one of the other synucleinopathies. And actually there is, you find even somewhat of a dose effect of the expression levels, meaning that quadruplication carriers would develop the disease earlier than triplication carriers or duplication carriers. So at least in familial disease, the disease progression and aggressiveness actually correlates well with the expression levels of alpha-synuclein. So since most of the talk will go about alpha-synuclein structure, I also kind of sum up some of the basics of alpha-synuclein. It's actually a very small protein, at least in its monomeric form, only 140 amino acids. It has a lipid binding domain, which is roughly the first roughly 90 amino acids of the protein, and an acidic tail, which is roughly 100 to 140 amino acids. And the C-terminus, those amino acids. It has a NAC domain, which is known to fold into beta sheet in disease and form these protein inclusions that are just cited as Lewy bodies. And it is shown to be involved in maintaining the plasticity of the synaptic vesicle pool. Well, so the protein is a little bit of a folding chameleon. So it does not only form one structure, it actually forms a lot of structure. And this is where my research is mostly focused. So I have here a citation of a review and just kind of borrowed their small diagram here. As I said, the protein, very small, but it does form a couple of interesting and physiological relevant structures. As I said, it's implicated in regulating the synaptic vesicle pool. And in that capacity, the protein likes to bind to lipid vesicles, fold into alpha helices. The protein also in disease likes to form aggregates that are also structured. They, again, as I said, they have beta sheet structure, and they form these long rod-like structures that ultimately are called or mature into amyloid fibrils, and those fibrils then actually make up the Lewy bodies that we observe in Parkinson's disease or dementia with Lewy bodies. Another form it has in the cytosol are just, if it's not membrane bound or aggregating, is a soluble monomer of the protein and also a folded soluble tetramer. Actually, we will focus today mostly actually on this equilibrium because it has very severe disease implications. So a couple of years ago, me and a couple of other groups discovered that the native protein structure of alpha-synuclein is actually not exclusively an unfolded monomer, as was thought to be the case previously, but that it also forms tetrameric structures that actually already are folded. So alpha-synuclein up until that point was seen as the flagship protein of an intrinsically disordered protein or a natively unfolded protein, which are proteins that are thought to have not any secondary structure per se, but adopt quickly whatever structure they need to bind to other proteins or to exert their physiological function. The interesting side finding of this particular study that we've done was that on a disease perspective, there were a couple of interesting things that actually followed from this. So obviously, very interesting topic would be why was this protein thought to be unfolded, and we've discovered that it is actually a folded protein in the human tissue context. And the answer to that is most studies beforehand actually characterized the structure from bacterial material, and we characterized it from actually human tissue, in this case human neuroblastoma cells and human erythrocytes. So question number one, what is different about human cells or mammalian cells? We've actually been able to replicate these findings in a large variety of mammalian cells compared to a bacterial cell. The second part that was immediately disease relevant, though, was that if the protein is already structured, we have to rethink a little bit how it aggregates, because the common assumption was that an unfolded protein will have a very easy segue into folding into a local protein folding minimum, like an amyloid fibril. If you just let it sit on the bench, people have observed that it forms these fibrils readily over a couple of days. And the question was more like, okay, what keeps it from aggregating in vivo? Now that we found that the protein actually to large part exists in an already folded structure, we had the application that actually it has to be destabilized first before it can go into an aggregation-prone version of the protein that can then fibrilize. And just to lift one part of the critical data from that paper was just, we did the experiment, let this guy, so the unfolded monomer, isolated from bacteria, sit on a bench, let the folded tetramer sit on a bench, isolated from human erythrocytes. And what we've seen with a thioflavine T assay, which measures pathological aggregation in this context, what we've seen is that the monomeric form of the protein slowly forms these disease-associated fibrils, while the tetrameric form here depicted in blue just stays stable and does not form or undergo these pathological changes, which was immediately interesting because it means, okay, what governs actually this equilibrium? How can we stabilize this physiological form, and how can we use this maybe as a biomarker? Because the ratio of unfolded versus folded protein should be usable in terms of showing a disease association. Going a little bit further, we wanted to nail down the disease association a little bit. So with my colleague Ulf Detmer, we did a couple of interesting experiments, just probing what do the familial PD mutations in the alpha-synuclein gene actually do to this equilibrium of protective versus aggregation prone form. Just because, again, we wanted to see, like, is it actually correlated with disease? So we knew those mutations do cause disease, and do they also cause this, we think, disease-associated change? And across the board, all of these 5 familial disease mutations actually destabilize the tetramer, which was a strong hint at that, yes, indeed, this would be the breakdown of the physiological form might be actually an early precipitant of the disease. It might be disease associated. We then try to do the reverse experiment. So if we think it correlates with disease, is it also able to cause disease? So we designed a couple of artificial mutants that break down or destabilize the physiological form, express them in neuronal cells, and we actually got the very nice result that yes, the more we destabilize the tetramer, the more we actually get neurotoxicity, and we get inclusion formation that does look a lot like Lewy body formation in these neurons, meaning that disease-associated factors are associated with the diminishment of the physiological tetramer, and if we artificially destabilize the tetramer, we get something that looks like disease-associated changes. The latest one in that was model system that Silke Nuber did and made in Dennis Selko's lab, and there she used these previously described mutants that artificially destabilize the tetramer, expressed them pan-neuronally in a mouse, and actually what she got was a mouse that by all accounts got something that looked like Parkinson's disease and was actually treatable with levodopa, just like a Parkinson's disease patient. Again, showing in a model organism that yes, if you just shift the equilibrium of alpha-synuclein forms to the less beneficial monomeric form, you actually do induce disease in the whole organism, which was an important finding. That all of these aggregated data led us to propose a new model of the disease, or not really actually that much of a new model, but more of an extension of the old model. So the old model, and I'm simplifying the model here quite a bit, started with an unfolded monomer, which was again presumed to be the physiological form of the protein, that then undergoes pathological changes into better sheet-rich aggregates, and that then forms these disease-associated pathological hallmarks, Lewy bodies. Most of the therapeutic approach and actually biomarker approaches were aimed at either detecting or blocking these species from happening. We extended the model a little bit, so if you look closely, you actually still see the traditional model in this part of our extended model. But we thought like, well, so our data implicates that before any of this can actually happen, we can stabilize maybe the folded tetramer, so this cascade doesn't actually even start, which gives us an additional therapeutical angle. And also, if any disease-associated stresses actually disrupt this equilibrium, we might be also able to harness the equilibrium of the physiological form versus the destabilized form as an early, again, hopefully a prodromal biomarker of the disease. Extending our options, stopping the disease in our mind a little bit later, we could address it in the very, very early stages here. For that, we also wanted to see translate our results into the human brain and actually, again, validate that this is true for human samples, human brain, not only model organisms, and that this is also true for sporadic disease, since so far in our model systems, we looked at genetic causes of the disease. For that, Laura De Boni, a very talented young neurologist in my lab, actually applied our cross-linking technique to assessing the monomer to tetramer ratio in a large amount of patient tissues across different diseases, so Parkinson's disease, dementia with Lewy bodies, and Parkinson's disease dementia, and first had a look at is the ratio actually diminished in sporadic disease. At the same time, we also purified the protein from human tissue just to make sure that it had the same properties that we previously recorded from human erythrocytes and human tissue culture neuroblastoma cells. And yes, indeed, it was also helically folded. It was also multimeric, and it was also aggregation resistant. And what we've seen is that these forms, once we isolated them and once we assessed their relative quantities, we saw not only are they decreased in sporadic disease, so if you compare here dementia with Lewy bodies or Parkinson's disease with healthy control tissue, it's very easy to see that the synucleinopathy patient brains have a diminished ratio of physiological aggregation resistant multimer versus aggregation prone monomer. But also one surprise that we had was we wanted to add an additional control. Instead of just having healthy control tissue or disease control tissue, we also wanted to probe different brain regions. And there we actually had a very interesting finding, namely that this diminishment of the multimer was region specific. So for example, here, if we show the data for the frontal cortex, which is has a heavy Lewy body burden in both dimensional Lewy bodies and Parkinson's disease, you see a diminishment of the physiological form. In other areas, which are affected actually by neurodegeneration, but do not have any Lewy body pathology, so their neurodegeneration is actually seen as a little bit secondary, of secondary cause, you don't have a diminishment of multimeric species. So this seemed to be just from those two regions associated with actual pathology in those brain regions, and if those are affected. We then went on and probed more regions, going basically from the brainstem through the midbrain to the cortices, so first frontal cortex and then occipital cortex, in the logic of, okay, which brain areas are affected the most and which are affected the least. And we also, for this graph, flipped the ratio a little bit. So the monomeric species, which we've seen as disease prone, we kind of feature here more prominently. So if you have a higher ratio here, so more of the disease prone species compared to the disease protective species, the multimer, you'll get a higher value there. And if you just probe different brain regions from the amygdala to the occipital cortex, all the way through, what you actually see is that you have more of the aggregation prone species closer to the brainstem. And the further away you go from what is seen to be the epicenters of the disease development, here shown for Parkinson's disease, actually the less you get of the disease-prone form of the protein, and relatively more of the protective form. So just to visualize this a little bit also, we drew a little brain diagram. Darker colors mean higher monomers or higher disease susceptibility, and lighter colors mean more relative protective forms. And as you can see like a gradient throughout the brain that gives you a certain pattern for, in this case, Parkinson's disease of these different ratios. Now, what we found very interesting is when we compared that with clinically used staging of the severity of Parkinson's disease, also known as Braak staging, and what is described in how the disease pathology develops throughout the brain. We saw a nice little overlap with what is described in these brain diagrams in terms of Lewy body burden throughout the disease. So a lot in the brainstem, a lot in the amygdala, then slowly radiating out to the midbrain and then to the cortices, together with our model of the aggregation prone forms of the protein, meaning that there was a direct link between where the form is destabilized and where actually disease develops in the different brain regions. Given that the Braak hypothesis always comes with the idea of spreading of pathology, we also ask a little bit of a different question here. So most of the listeners probably will be already familiar with the Prion hypothesis that kind of followed Braak's staging scheme, but also to kind of give a brief overview here. So how the disease pathology or the development and spreading of disease pathology through the brain, different brain regions, is thought to be happening is actually through small misfolded protein particles of alpha-synuclein that literally diffuse through the brain tissue, get taken up by cells, and then induce actually aggregation of the endogenously regionally produced alpha-synuclein in that particular brain region. Mostly those findings were kicked off A, by the Braak staging, and then B, by actually a couple of papers in Nature Medicine in 2008. which found that patients who received as a potential treatment stem cells into areas of neurodegeneration. So those patients got stem cells into the substantia nigra injected in the hope that the stem cells would locally differentiate, produce dopamine, and rescue some of the phenotype of the disease. What was clear after this long-term experiment was, though, that autopsy of those treated patients also revealed that Lewy body pathology was found in the newly grafted neurons and not only in the old neurons, implicating that actually the disease is not necessarily a function of time per se. So it's not something that happens all over the brain at the same time while you age, but it is something that develops in one cell and actually can jump to a much younger cell and quote unquote infect the cell, because the newly grafted cells were obviously very young material, while the surrounding brain was very, very old. So if it's not really infectious, if it can't really jump from brain cell to brain cell, one would have expected the newly grafted neurons to be fine, but Lewy body pathology was found. So the implication was there that the disease is literally spreading from cell to cell. He tested this particular prion spread also with our tetramer and monomer ratio. So I've already shown you, and we've published in the past, that spontaneous aggregation is something the tetramer is completely resistant to. So it doesn't just on its own develop into these amyloid fibrils. The monomer does. What we also now found interesting, what happens actually if we mix both the monomer and the tetrameric form with these prion species of alpha-synuclein, which are able to convert physiological protein into pathological protein. And the surprising finding there was that yes, the monomeric form actually is susceptible to the prion conversion. So if you mix small amounts of the prion form of alpha-synuclein with monomeric form of the protein, it will greatly enhance aggregation and accelerate aggregation, while again the tetramer seems to be completely protected against prion changes. So you can use as much prion alpha-synuclein and mix it with the tetrameric form of the protein, but it actually won't aggregate even under these conditions. To get this finding a little bit more robust also in a cellular system, we thought, okay, we know that in a cellular system we have a certain amount of tetramer to monomer ratio, and we can actually tweak that ratio by expressing different mutants. As I already told you, the familial PD mutations, for example, all destabilize the tetrameric form of the protein and relatively enrich for the monomeric form of the protein. So if destabilization of the tetramer also in cells would lead to higher susceptibility to prion, what we should actually see is that if we put prion alpha-synuclein into the cell medium of cells expressing various mutants, they should be actually susceptible to the prion aggregation in a manner that correlates with the destabilization power of the tetramer. So what we've done here is we expressed all the 5 familial PD mutations and then also 2 compound mutants, one called 3X, which combines the H50Q, G51D, and E46K mutation, and one 5X compound mutant, which actually combines all of the 5 familial PD mutations in one gene. We expressed them, treated the cells with prion alpha-synuclein, preformed fibrils, and just measured after 2 days how much insoluble alpha-synuclein do we get in these cells under identical expression conditions. What we saw easily was that relative to the total amount that those cells expressed, the mutants were more susceptible to prion expression, and the compound mutants were actually far more susceptible to prion expression. So if we just measure the amount of insoluble alpha-synuclein after 2 days that we can record in all of these cells, and just plot it on a graph together with how much they destabilize actually the physiological tetramer and lead to an increase of the aggregation or prion-prone monomer, what we see, we get a very nice correlation with the severity of a mutant in terms of how much it actually destabilizes the tetramer, and then also how much insoluble alpha-synuclein it generates under prion seeding conditions, meaning how susceptible is it towards prion-like spread. So under these aspects, we can now understand why the Bragg pattern is the way it is. Previously, we were actually a little bit puzzled why the Bragg pattern spreads through certain regions faster than through others. But our finding of this pattern of more relative monomer versus the protective form, the tetramer, actually might give us a first clue here. Because if you think about that the monomer is more susceptible to prion spread, then obviously the prion, once it actually starts, would spread through regions which have a relative high level of monomer first, and then spread through tissue that has a very low level of monomer last. Only if the prion burden gets very high, those regions will be affected. And that is actually what we saw. So if we assume that the prion spread actually starts more towards the brainstem, so we took here the amygdala, which is a little bit further removed from the brainstem, but still close enough, as a first starting region. What you can see in PD is if you follow the multimer to monomer ratio, again, multimer being the protective form, the prion actually here has a little bit of an uphill battle to fight. So it would spread slower and slower through these regions towards the cortices, meaning that you would get very early brainstem involvement, and only as the prion load builds up, you would get affected midbrain and then only the cortical regions, which was very interesting because that actually also mirrors the clinical development of the disease, where you have actually one of the most prominent clinical phenotypes is midbrain involvement, so movement phenotype, and actually dementia comes fairly late. Interestingly, dementia with Lewy bodies has a very early, so another synucleinopathy has a very early dementia phenotype. It also has a movement phenotype, but the movement phenotype rapidly actually subsides to the dementia phenotype. And when we actually graph the same pattern over specific brain regions for dementia with Lewy bodies, and I don't have the data on here, but also for Parkinson's disease dementia, we also have a very early involvement of the cortices. You can see actually that prions here would have a downhill effect. So they would have basically a highway that would lead them towards the cortices, meaning that if prions develop in the brainstem or amygdala here, they would actually don't need to build up that much of a load before they can just march through to areas where they have lower amount of multimer that would make them more susceptible to prion spread. So this pattern not only is an explanation for Bragg staging or the pathology spread in Parkinson's disease, but it might actually also explain the spread in the different diseases and also the clinical phenotype in both diseases. So switching gears a little bit, one of the interesting questions that obviously now comes out of this is, okay, so this is nice. So we have the regional specificity of a multimer to monomer ratio that kind of gives us a little track map of how a prion might actually spread through the brain, and that is consistent with the clinical phenotype. But what actually governs the multimer to monomer ratio? Because obviously the therapeutic target would still be gaming that ratio a little bit, because the implications also reverse that if you stabilize the multimer, you would actually protect a certain brain region from pathology. And there I want to kind of sidetrack for a second and describe a paper that was recently published last year, stating that alpha-synuclein inclusions, aka Lewy bodies, not only exclusively consist of amyloid fibrils, but they also consist of alpha-synuclein lipid clusters, showing that aberrant lipid interaction of alpha-synuclein is actually one of the main hallmarks of the disease, which was very interesting to us because one side finding I haven't gone into in my intro about my previous work with Vesicle was that these artificial mutants that we expressed first in neurons, and then Silke-Nuber made a mouse model out of them. One side finding that they had, not only did they destabilize the tetramer and led to more relative monomer, but across the board also those mutations led to higher amounts of alpha-synuclein bound to lipids. And actually some of these lipid inclusions that we described in an early paper in 2015 turned out to be also just aberrant lipid clusters in a paper that we published later in 2017. In addition to that, we have actually also shown in a couple of in vitro experiments that lipids are able to convert monomeric protein into the stably folded physiological tetrameric form. So a very talented student in my lab in 2018 developed a system, an artificial in vitro system where we could mix alpha-synuclein with lipid vesicles, which it's known to bind. And we could, through choosing the conditions carefully, actually switch on and off alpha-synuclein binding to those lipid vesicles. Now, as I described to you before, lipid binding of alpha-synuclein has been described before, also in vitro. What's never been actually looked at has been what happens after alpha-synuclein has been in contact with lipid vesicles. So what has been done is just showing alpha-synuclein in vitro without any vesicle contact doesn't have secondary structure. If you bring it in contact with a lipid vesicle and it binds, it does form helices and it binds to the vesicles. But again, we did the next step and then asked, okay, what now if you switch off the lipid binding? What happens to the protein? Does it come off folded, or does it stay in its fold and all of a sudden become cytosolic and folded? And lo and behold, in that paper, we could show that, yeah, by switching on and off alpha-synuclein lipid interaction, we could actually convert unfolded monomeric protein into helically folded multimeric protein that was also not bound to the vesicle anymore and cytosolic, meaning that the lipid vesicles, just the temporary contact, actually functioned here as a liposomal chaperone of some kind. So lipid interaction is not only part of the pathological phenotype, but it is also apparently an important puzzle in determining, okay, how much tetramer versus monomer do you have? Because for this model that I showed you on the last slide to work, actually we need alpha-synuclein to bind to vesicles, but then also to come off of vesicles a little bit, which it does in vivo all the time. So it has been described that it actually comes on and off lipid vesicles. That seems to be tied to its function in regulating synaptic vesicle plasticity in vivo. Roughly, if you take a snapshot, you have 15 to 20% of alpha-synuclein always bound to vesicles, but that doesn't mean that 20% of that protein are stuck to the vesicle, but it just means it's a very fast exchange in a snapshot, which is captured the 20% that happen to be at that millisecond on the vesicle. We wanted to also now investigate our brain tissue a little bit. So is that something that we see? Do we also see some early hints at aberrant lipid interaction? Because combining our finding that you need alpha-synuclein to touch a lipid vesicle but come off again to form the tetramers, and the pathological finding that alpha-synuclein in disease actually is stuck to a vesicle would imply that actually if you have too much alpha-synuclein on lipid vesicles, not only does it impair lipid vesicle trafficking, but it also will disrupt tetramer formation. So we looked at the brain tissue, and we looked also at different pools of alpha-synuclein. We wanted to investigate a little bit, okay, how do these pools of alpha-synuclein in the cytosol, membrane-bound, and insoluble change over time? And time here is given by Braak staging, which again is neuropathological staging of disease progression, meaning Braak staging 3 or 4 would be somewhat early in the disease, while 5 or 6 would be actually very late stage of the disease. So once we investigated a couple of brain areas, now that aspect, what we actually seen is that the soluble form of alpha-synuclein, as expected, throughout the Braak staging, so through progression of disease from Braak 4 to Braak 5 to Braak 6, actually goes down. That is not surprisingly that the soluble form goes down, because I just told you that pathology is associated with more insoluble protein. So we just thought, like, okay, so less soluble protein because it aggregates into amyloid. What we then saw, though, when we separated insoluble amyloid alpha-synuclein from just membrane-bound alpha-synuclein, we saw that the decrease in cytosolic alpha-synuclein is actually much better mirrored in increase in membrane-bound alpha-synuclein throughout the progression of the disease. And that was a feature of all the brain regions that we looked at. So there was not a region-specific pattern, but here there was a temporal pattern actually observed. So the worse the disease gets, the more alpha-synuclein you have on membranes. And with that, to make the story also just a little bit more complicated, I just showed you that lipids can work protectively to fold alpha-synuclein into prion-resistant species, but it also has been shown by other labs and also in our lab that lipids, if alpha-synuclein binds too long to those lipids, might actually also provide a hydrophobic surface to aggregate the protein. So too much lipid interaction might not be only a problem in terms of preventing tetramer formation, which makes the cytosolic protein aggregation resistant, but it actually might kick off aggregation itself just because alpha-synuclein accumulates on a hydrophobic surface. As the Dobson group, is the paper that I'm showing here, as well as our own data has shown that if alpha-synuclein binds too much to lipids, that is actually another factor that accelerates aggregation. And this brings us to an even more complex model of aggregation. So here I actually put out the part about the tetramer formation. So at this point, we already have destabilized tetramer. But what this also says is that we should, on the other hand, not only look at cytosolic alpha-synuclein, how it in the cytosol aggregates into fibrils, but we also should definitely take into account that alpha-synuclein interaction with the lipid vesicles itself can kick off aggregation into Lewy bodies, and that might be actually what we observe in the disease. That leads also to a very interesting idea that not only do lipids maybe accelerate alpha-synuclein aggregation and determine if it falls into tetramers or how long it actually resides on the vesicle, but that also might mean that the exact composition of a vesicle not only determines the speed of alpha-synuclein aggregation, but also that it might actually kick off the protein in different forms that exert different bioactivity. So one of the factors or one of the roles that lipids might play is actually determining if alpha-synuclein goes into harmless aggregates, into toxic aggregates, into prion-like aggregates, and even in the whole prion spreading, if it converts into forms that spread more into a DLB pattern or a PD pattern or a multiple system atrophy pattern. Okay, and that with that model, I will close down and thank all my collaborators over the years, especially highlight. I want to do Laura De Boni, who did most of the human brain work that I showed you here, but obviously all the current and past members of my lab and all the collaborators that I've worked with on this project. And of course, as always, the funding bodies that made all of this work possible. Thank you very much for your attention.